The overall goal of this procedure is to record odorant-induced responses from mouse olfactory receptor neurons. This is accomplished by first dissecting out the olfactory epithelium and isolating the mouse olfactory receptor neurons. The second step is to place the isolated olfactory receptor neurons in a recording chamber mounted on an inverted microscope. Micromanipulators hold the recording electrode and a solution changer. Next, suck an identified olfactory receptor neuron into the tip of a suction pipette electrode. The final step is to place the pipette with the sucked cell in its tip in front of a solution changer to expose the neuron to odorants. Ultimately, the suction pipette recording method is used to investigate olfactory receptor neuron function.
The main advantage of this technique over existing methods like whole cell voltage clamp technique is that it allows for long recording durations using fast and precise solution changes.
Visual demonstration of this method is critical as the shape of the recording pipette is important, and placing the sucked cell stably in the tip of the pipette is difficult to learn.
Begin this procedure by pulling a micropipette with an unfilamented borosilicate glass capillary in a micropipette puller with a long taper. Then transfer the pipette to a custom-made micropipette holder mounted on an upright microscope. The microscope has a reticule in the eyepiece and a movable diamond knife mounted on the stage. Next, observe the tip of the